Hi, and welcome to the Miltenyi Biotec session. I'm Amy JABLONSKI, the Technology and Application Specialist with the Imaging Group, and I'm happy you stopped by to hear about our imaging solutions and their applications. So let's get started and dive into Explore Deeper, from whole mouse to tumor microenvironment. There is no mistaking that understanding the 3D environment in biology has become an important aspect for many scientific questions. For example, in neuroscience, the disciplines of brain imaging, nervous system imaging, and neuron activity analysis have benefited from 3D microscopy imaging tools and markers. In the past, these applications would need to be sectioned to be imaged, thus losing the 3D spatial information. However, with volumetric imaging techniques, this research can now obtain detailed structural mapping and connectivity tracing. So whether it's understanding the connectivity of an entire whole organism like seen here on the left of this 3D reconstruction of neuronal projections in a mouse, or as on the right, the vascularization of a whole mouse brain, 3D imaging has played a big part in understanding neurodegenerative diseases and neuronal plasticity. 3D fluorescent imaging is also fast becoming important in immuno-oncology and for assessing disease progression and drug efficacy. Drug discovery assays have seen the benefit of imaging spheroids in 3D to better understand, say, how a specific drug compound affects the spheroid, including drug penetration and interaction with its environment. In tumor biology, 3D imaging on an even larger scale, such as that of xenografts, can give insight into cell localization and even quantify tumor volume. In this video, we explore xenograft of a human pancreatic carcinoma cell line, shown here in green, in addition with the infiltrating CAR T cells represented in magenta. The volume rendering allows for the investigation of the interaction of the CAR T therapy with the cancer cells in its native environment. The 3D spatial interactions of the cells within the tumor has become crucial to understanding many aspects of tumor biology, including that of tumor growth, progression, prognosis, and treatment avenues. It's no longer that specific cancer cells are present, but also the interaction with its environment, such as immune response of the host system and the role of the extracellular matrix. While tumor cells are definitely a key constituent of tissue masses, we're also starting to realize they're surrounded by a complex microenvironment. Each one of these cell populations that may be present actually in the tumor environment consist of their own cellular subtypes at various abundances and spatial locations. It is these differences in protein abundance and localization across a macro and microscopic scale and tissue that actually may contribute to, say, the unpredictability of some cancer treatments or patient-to-patient differences. Thus, this is the need for appropriate technologies and techniques to understand the multiple facets of this tumor microenvironment. Here at Miltenyi, we recognize the need for a range of imaging solutions to tackle the scientific questions related to spatial interactions, whether this be in the tumor biology space or neuroscience, or across a wide range of macro and microscopic scales. Now, I refer to them as solutions as it's not just the instrumentation, but also the whole workflow, so that the researcher can quickly get to the answers that they are looking for. Now, while we have multiple different imaging solutions, I'll be focusing on the light sheet and the ultra-high content imaging solutions and their applications, and how they brought light to a complex tumor environment from all different perspectives. So let's jump right in with the light sheet microscope that is focused on high-resolution 3D imaging. While many of you already have experience with light sheet microscopy, I'll just briefly touch on the technology. Let's take this zebrafish containing fluorescent molecules as an example. With light sheet microscopy, the excitation and the detection pads are uncoupled and oriented perpendicular to each other as represented by these two objectives. The excitation beam illuminates the sample from the side, and detection of the fluorescence occurs on the vertical axis. The nice thing about this is that the excitation beam can propagate through the width of the sample, and therefore the light sheet itself is creating the optical section. This means there is no requirement for blocking out-of-focus light with a pinhole or amplification of signal with PMTs. Instead, the light sheet microscopes are able to acquire the entire field of view in a single camera image, as you can see here on the right. Thus, when combined with tissue clearing, light sheet microscopy allows the imaging of a large range of sample sizes. If we look at the graph here of kind of the range of all the biological samples that you may be interested in looking at, not only are we able to image, say on the left side here, those smaller samples such as Drosophila brains and organoids, in which you probably may have already used confocal workflows and have gotten great results with. But also, we can actually work our way towards the right there, these much larger organisms and organs that you would be interested in looking at. This is where confocal usability usually decreases, but the light sheet actually starts being able to use to really get into those larger samples. This brings a series of advantages to using light sheet microscopy, including first, decreased photobleaching, as the excitation beam propagates through the width of the sample to create the optical section of interest. Second, as you saw in the previous slide, combining light sheet microscopy and that with long working distance objectives and that tissue clearing really allows for the wide range of samples that otherwise would need to be physically sectioned. Lastly, with no requirement for blocking out of focus light with a pinhole or say scanning across a field of view to acquire signal with PMTs, the light sheet microscopy can achieve faster acquisition times while still being able to resolve individual cells. This has made light sheet microscopy a method used across various fields of science such as neuroscience and immuno-oncology to visualize large 3D samples, organs, and small animals as seen in these covers. Now, those of you that have done light sheet microscopy know there is more to it than just imaging the sample. To achieve those types of images, there are a couple of considerations in sample preparation and imaging conditions. And this is where Miltenyi Biotec likes to take, say, a holistic approach. Instead of just thinking about the instrumentation that you're taking the images with, thinking about the whole light sheet workflow, so that we can reduce the barrier of researchers being able to work with light sheet microscopy in their research. And in doing so, we're allowing the researcher to focus instead on their specific scientific outcomes. Now, to begin, one of the biggest challenges when staining thick tissue sections or entire organs is the complete penetration or diffusion of antibodies through the entire sample. To ease this burden, we have 3D immunofluorescent validated antibodies that have been screened for functionality and optimized for tissue penetration. You can observe this with the image on the right. Here, a cerebral organoid was stained with beta-3 tubulin. In the case of the Miltenyi Biotec 3DIF antibody, this antibody has penetrated deeper into the organoid, reaching the center, so that distinct features can be seen, whereas if we compare this to the image on the left, these features look impaired. These recombinantly engineered reaffinity antibodies, directly conjugated to bright and photostable VO dyes, offer many advantages of direct immunofluorescent staining, including reduced staining time, great signal to background ratio, and minimal background signals as compared to indirect staining. The next consideration is tissue clearing, which I believe you guys have heard me talk a lot about at the beginning of this talk. If we consider normal, live, or fixed tissue, there's many different materials within the tissue, such as water, proteins, and lipids that really impair the light passage. As seen in this simplified view of tissue, these materials that the light encounters causes things like reflection, refraction, scattering, and absorption, and decrease the light penetration, detection, and then ultimately the image quality. So the goal of tissue clearing is to basically render the tissue transparent, like we see in this movie, reducing these impairments so that we get better imaging. Now, specific protocols have been developed using the Max Clearing Kit for a easy, fast, and effective immunofluorescent staining and clearing of different tissues. However, it should be noted the system is not limited to only using this kit and has the flexibility and the support to handle all different types of clearing methods, ones that even you have maybe already worked with and have optimized for your specific samples. Last is the instrumentation to visualize this, right? We've all done this work up front to be able to now get those really nice, images out in the end. So the combination of our cutting edge light sheet illumination technology and objective lenses ensures an unprecedented image quality across scales to create a 3D volume of your sample. Now, there are some unique features that I'd like to dive deeper into that really gives this scope its ease of use, flexibility, and quality of imaging. Here is an image of the ultramicroscope blaze. The first thing that I'd like to point out is the cutting-edge light sheet optics. The ultramicroscope is equipped with 6 light sheets, 3 on the left side and 3 on the right. As you can see in this little image on the right-hand side, this really gives the scope this homogeneous illumination, which in turn is able to give us improved sectioning and ultimately the superior image quality that we've seen in some of those images before. The second thing is the system is equipped with 3 long working distance lenses that reside on the scope. There's no need for the researcher to change these out to decide on which is best for their sample. And this system also has an intermediate lens exchanger for applying deep magnification and post magnification capabilities to a particular objective of 0.6x up to 2.x. This really gives you more versatility out of each of the objectives that you have on the system. Additionally, these objectives are fully automated. As you can see in this video, you can easily switch the objectives at the touch of a button. And not only that, is that you can automatically then find the sample to really streamline the imaging process. And last but certainly not least, is the large automated sample chamber for easy access and mounting of multiple samples. We're going to actually look at this a little bit closer in the next slide. At the touch of a button, you can safely release and mount the sample into the large imaging chamber. This could indeed be a whole mouse, or you could mount a series of organs as we've done here. The idea being, though, is that once this is loaded into the system, a batch acquisition can be set up to automate the acquisition of all of these samples. So this really allows the researcher to load it once, set up a few parameters, walk away, get other things done, and then come back to look at their images, say, the next day, and really focus instead on the data. So let's examine some of the scientific questions that one can tackle with this workflow. We can begin with something on the scale of a couple hundred microns, like this brain organoid. In this example, we can see the neuronal marker of beta-3-tubulin in cyan in combination with Ki67 in yellow and SOX2 in magenta. So as we take a look, we can then section through to see through the entire brain organoid and be able to really get down to those individual cell levels. But it doesn't only have to be cells derived from a mouse. It can certainly be a whole cleared mouse brain. The example you see here, researchers used immunolabeling, light sheet microscopy, and deep learning computational methods to get a comprehensive and non-biased map of MPTP-induced changes in the central dopaminergic pathways important in Parkinson's disease. Now, this really allowed them to create a full model to get a complete three-dimensional visualization and then also automate the quantification of the tyrosine hydrolase positive neurons in the adult mouse brain without having to section the brain. We can even go into whole mouse heart. Here, researchers were looking at the restoration of perfusion after acute myocardial infarction. This is the most frequent and effective medical treatment, but the process may inflict ischemia reperfusion injury, or IR injury. Now, a hallmark of this injury is the loss of vasculature and the extensive inflammatory response. Thus, by using 3D analysis, this really allowed the researchers to simultaneously characterize the extent and also the localization of the damage and the ensuing inflammatory response in the whole heart. Now, as you're watching this movie play through, you can distinguish the acute and late vascular IR damage zones. These also contain highly localized and spatially structured neutrophil infiltrates. These were represented by the Ly6G positive cells and spots that are modulated upon cardiac healing. And here in mouse lung, the researchers were investigating human innate lymphoid cells, ILCs, in the context of mucosal inflammation. By using light sheet microscopy, they could better understand the ILC2 lung homing mechanism in cystic fibrosis lung pathology. In this representative animation of the 3D reconstruction of the lung lobes, you can see the lung accumulated human ILC2s in red after a 24-hour IV transfer into a mouse with the papain-induced lung inflammation. It is also possible to move up to imaging a whole mouse. Yes, a whole adult mouse can be cleared and mounted into the ultramicroscope blaze. This is actually one of my favorite examples of the power of the light sheet microscopy to understand the tumor environment. In this example, researchers were looking for a reliable detection of disseminated tumor cells and of the biodistribution of tumor-targeting therapeutic antibodies within the entire body of this mouse. This would allow them to better understand and then hopefully treat cancer metastasis. Here, the researchers used a combination of light microscopy and an integrated pipeline for automated quantification of cancer metastasis and therapeutic antibody targeting, named DeepMap. Now, as we explore this mouse, we can see a distribution of sizes of the tumors, shown in magenta. While there are definitely some larger tumors present, there are also these micrometastases of various sizes across the entire body. Now, an interesting finding here was that after treatment with a targeted antibody-derived drug, there were a number of untargeted micrometastases. You can see that now here where the targeted tumors are actually in white, but there are also a lot of these still untargeted tumors in magenta. The researchers ended up extracting these micrometastases, serial sectioning them, and then using a confocal to get a deeper look into the vessel location and size, but also the co-clustering analysis of the targeted and untargeted microtumors, hoping to understand what was the difference between those that actually were targeted by the antibody treatment and those that weren't. The conclusion is that there was the existence of factors in the tumor microenvironment that were influencing the efficiency of the antibody drug targeting. And so if we think back to this beginning slide talking about the tumor microenvironment, you can imagine how daunting the task would be to understand all those factors. In their case, they looked at a couple of things and weren't able to pinpoint it. So if we really wanted to be able to explore all the different cell types and subcell types present, you can quickly see how you might need more than 50 markers to even fully characterize the tumor microenvironment to answer a question like that. So there has been some work on this, and in order to better characterize a tumor microenvironment, there have been methods that have worked on to get those high number of markers that you would need for something like that. However, most of the time there is a trade-off between high number of markers or spatial information. Flow cytometry has been able to use a high number of markers to analyze a sample, but the tissue usually has to go through a dissociation process, losing all that spatial information that we would want. In contrast, immunohistochemical methods keep the tissue organization. However, whether using chromogenic or fluorescent methods, there is a limit to the number of channels and markers that can be used, and getting the magnitude capable that we have on flow, the user would need to take a number of serial sections of the tissue. Thus, the push for higher multiplexing techniques that would preserve the spatial information of all the markers needed is definitely there. There are many ways to approach multiplexing. In our case, our approach is based on immunofluorescent cyclic staining, which we call maximal imaging cyclic staining, or MICS for short. In this approach, we stain the prepared tissue with up to 3 primary conjugated fluorescent antibodies, and that's done within our system. Next, we will sequentially image those antibody markers along with DAPI for the nuclei. And then eventually we will erase the fluorescent signal via photobleaching or enzymatic release. And this is done before automatically starting the process all over again until the desired number of markers are reached. And as you can see, as we cycle through all of those markers of the colon carcinoma sample, is that the result is the capability to build up a final image with hundreds of markers, while also containing all the spatial information on that single piece of tissue. The mixed technique has a couple of advantages over the current techniques. First, as you saw on the previous slide, it allows the labeling of hundreds of markers on a single piece of tissue, eliminating the need to do serial sectioning that may not always be possible when the tissue is scarce. Second, this technique retains the spatial information of the cells that is lost when you're doing high parameter flow cytometry techniques. Third, this is a non-destructive process. Unlike other methods that you may need to ablate or disturb the tissue, this process keeps the tissue intact and could potentially be used for downstream applications. This is all brought together with the Maxima imaging platform as a whole multiplex solution. First is the fully automated instrument with integrated imaging and onboard staining, so that after adding your sample and reagents, the researcher can just press play and get on with their day, leaving them instead to focus on the data that comes out from the run. Second, the adapters that let them image almost any sample they can think of, whether this is prepared tissue section, tissue microarrays, or even cultured cells. Simply snap these on and place in the instrument to do all the staining and imaging there. Third is the extensive antibody portfolio that allows detection of more than 400 antigens on human or mouse samples. Based on Miltenyi Biotec's recombinantly engineered reaffinity and release technologies, these antibodies are validated in-house for the specific staining conditions and a variety of fixation methods, including acetone, PFA, and FFPE methods. This is not to say other antibodies cannot be used inside the system, but by having these validated antibodies, we hope to ease the burden of the researcher to have to test a slew of antibodies, and this allows them to easily scale up with the complexity of their research question. Last, once the data of all those markers is collected, understanding how it all comes together can be a daunting task. With a MaxIQ image analysis software, you can do so much more than just visualize your markers. Indeed, from the image, you can actually easily identify individual cells and be able to extract relevant cell and biomarker-based measures. You can use these cell identification and biomarker measures to be able to classify certain cell subpopulations. And not only that, is once you have that done, you can also start extracting data and displaying this in a form so that you can better visualize what you're seeing. This is not limited to, but includes t-SNE plots, heat maps, and distance maps. Now, the great thing here is that this is all contained in one software, and that all that spatial information is retained through this process. So if you were interested in looking at a specific cluster or heat signature, you can always bring that information back to the image that you have. Now, in the following example, we'll show you how this all comes together and the power of the analysis software. So let's go back to that example that we saw cycling through at the beginning of this section. By examining tumor-infiltrating leukocytes, or TILs, in colon carcinoma. Colon carcinoma is one of the most common tumors in the digestive tract, and its tumor microenvironment determines tumor germination and progression. So understanding the presence of TILs in this environment will play an important role in tumor cell killing during immunotherapy. Now, here we can definitely understand the relationship of the TIL to the tumor cells using traditional imaging techniques. And it would look something like this, in which we could have the tumor cells labeled in green, leukocytes in red, plus blood vessels in blue, and then also the fibroblast. However, if I needed to dig deeper into this and actually understand the other cell populations that were present, we can look at this schematic on the right-hand side. And if I wanted to dive deeper into the different subtypes of leukocytes, I may need as many as 3 to 4 markers to even identify one of those subpopulations. And that's where multiplexing really starts to play a big role. Here we can again see just our 4-channel image, which we can definitely get some information out of. But by doing multiplexing, we can convert it into something more like this and be able to break down to those major leukocyte populations. So within all of these images, we've got the tumor labeled in green again, but now I can start breaking down that leukocyte into different things like CD8 positive cells, CD4, look at different Treg cells or natural killer cells, all because I'm able to stain on this one piece of tissue. And because the images of these cells retain all that spatial information, with automated analysis, you can really start selecting the infiltrating leukocytes versus the non-infiltrating ones. So if we look at the image on the left, we can see now the tumor cells are in red and the leukocytes are in cyan. And that if we take a deeper look into this, based on the location of where these are, you can start isolating the ones that are in close contact with the tumor, which would be present here in green, or those that are not interacting with it, which are in magenta. After isolating each population of leukocyte, the expression profiles of all the markers were plotted in order to identify markers of interest that are differentially expressed in infiltrating and non-infiltrating leukocytes. So if we look at the heat map here, we've got the two different populations of infiltrating leukocytes and non-infiltrating leukocytes. These were then clustered into 5 groups by k-means clustering. And then if we look at each one of those markers, so each column is a different marker, we can compare those expression profiles between those two populations. And what we were able to find is that 7 markers were selected, which are displayed here in red rectangles, based on the different expression profiles in this heat map here between the two populations. And it's not only that you have the expression profiles and you can get a heat map of the data out, but that because we retain the spatial information, you can then also go back to the images to look at what these markers look like. So here we have overlays of the different markers with the tumor cells and in comparison to those that are expressing CD3 and also CD8. And so in this case, the researchers found that markers 1, 3, and 4 were the most promising to be able to do further experiments and analysis with downstream. That is only one example of the power of the maxima and the mixed technology. We have a variety of other applications, including looking at tertiary lymphoid structures in ovarian cancer, using the mixed technology in CAR T-cell therapy discovery, and then also classifying and characterizing glioblastoma formae. And with that, I'd like to thank the organizers for the opportunity to talk, and thank you for visiting our session today. If you're interested in learning more, please be sure to check out the materials and the videos in our virtual booth.